Hello everyone, I am Wen Shen. I am an assistant professor at the Mechanical and Aerospace Engineering and the Nanoscience Technology Center at UCF. I joined UCF in summer of this year, and before that I was an assistant professor at the University of Texas at Arlington. Today, I would like to introduce my work on biomimetic sensors and applications. Okay, here my lab is called the Functional Materials and Devices Laboratory. In our lab, our research is focused on the design and the development of sensors and devices based on functional materials. First of all, I would like to introduce a case study on our development of biomimetic devices for neural interfaces. For neural interfaces, most of the times people use a device called neural microelectrodes. These electrodes can be used to record brain's electrical activities. They can also be used to treat diseases and release symptoms using electrical stimulations. In addition, engineers have been able to use these devices to reconnect brain's activities to the peripheral nervous system's activities. And by using this connection, engineers are able to build prosthetic arms and legs to help patients who lose functions in their arms and legs. However, regardless of these applications and their successes, a grand challenge for this type of neural interfaces is the incompatibility between the devices and the surrounding tissues. The incompatibilities are in the two different fields. One is the mechanical mismatch between the device and the tissue. Biocompatibility is another issue at the interface. Devices will be recognized by the tissue as foreign bodies, and a series of foreign body reactions at the device-tissue interface will also result in the scarring formation, and therefore the signal-to-noise ratio and sensitivities of the devices will also be degraded. To solve these challenges, we have been trying to develop neural interfaces using natural materials such as extracellular matrix materials or ECM materials. ECM materials are a group of materials that are secreted by cells to form their environment. We're hoping that by using these ECM materials to make the neural interface devices, we will establish an interface that can be more compatible to the tissue than conventional devices. However, a challenge is how can we create these transducers and the electrical interfaces from these ECM materials. Traditional silicon fabrication are not compatible to these biological materials. We have developed a series of fabrication methods that enabled us to fabricate ECM materials into micro sizes. Here you can see a comparison of commercial neural nexus electrode with the ECM microelectrode that we developed. You can see we can actually make ECM neural electrodes with similar sizes as the commercial neural nexus electrodes. Here is a set of fabrication methods that we have developed to make these ECM-based neural microelectrodes. We used magnetic particles as the mold to define the shape of ECM hydrogels. After that, we have used multiple times the transfer molding method to encapsulate the electronic part into ECM hydrogels. And then we align these ECM hydrogel sheet onto a stainless steel stent, which is used as a laser ablation mask and also as the insertion tool for these electrodes. These ECM microelectrodes exhibit adaptive mechanical properties. When they are dehydrated, their Young's modulus are relatively high, whereas when they are hydrated, the Young's modulus of these devices can be reduced by 1,000 times. The implantation of these devices is also affected by the implantation speed. The higher the implantation speed, the easier it is to implant these devices because the load that is needed for the implantation is smaller at a higher implantation speed. We also looked at the biocompatibility of these ECM bioelectrodes. Here we show that ECM bioelectrode can allow neurons to grow on them, whereas the electrode made from synthetic materials does not allow neural growth without any adhesive coating. And we also looked at early stage of our chronic studies, and we found that the ECM bioelectrode does not show very significant scarring around the interface. We have designed carefully the electrical properties as well as the electrochemical impedances of these ECM bioelectrodes, and we're able to record neuron activities using these ECM bioelectrodes. This technology is not limited to neural interfaces. It can also be used to make wearable devices. For example, we can fabricate wearable sensors on the ECM substrate. In addition to the development of sensors and devices using biological materials, I've also like to introduce the development of sensors and actuators using giant magnetostrictive or GMS materials. This is a type of materials that can convert magnetic energies into mechanical energies and vice versa. I have developed a detection system which enabled us to interrogate with micro-sized scale GMS sensors with improved signal-to-noise ratio, improved sampling rate, and also without the need of biased magnetic field. By using this detection system, we can develop biosensors to detect foodborne pathogens directly on the food surfaces. We can also develop GMS sensors for wireless and in-situ structural health monitoring applications. We can embed these GMS materials into composite materials, and then we have developed a wireless detection system to interrogate with the sensors that are embedded in the composite materials or structures. We're able to detect the quasi-static strain change as well as dynamic strain change in the composite materials. In summary, I have developed biomimetic devices for physiological interfacing and for agricultural and environmental monitoring, as well as for structural health monitoring. I'm looking for collaborations in the following field, and my work at UTA was funded by USDA, and my previous work was funded by NIH, DARPA, and SF. Thank you very much.